Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Personalized Cancer Medicine Needs Randomized Trials. My name is Diane, and I will be your X-Talks host for today. And today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. And this webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit any questions or comments you may have for our speakers throughout the presentation, and you could do so by using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any other assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode, and please note that this event will be recorded and made available for future download. At this point, I would like to thank IDDI for the content of this webinar. International Drug Development Institute is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Raleigh, and San Francisco. And now I would like to introduce our speakers for today's webinar. Our first speaker is Dr. Everardo Saad. Everardo is the senior medical expert and has over 15 years of experience in medical oncology and clinical trial designs. He graduated in medicine and trained in internal medicine in Sao Paulo and did his fellowship in medical oncology at the University of Texas MD and Cancer Center in Houston. And our second speaker for today's webinar is Mark Boyce. Mark holds a Doctor of Science in Biostatistics from the Harvard School of Public Health in Boston, and he is the founder of International Drug Development Institute and of CluePoints, two biostatistical service organizations based in the US and Europe. And now, without further ado, I would like to hand over the mic to our first speaker, Everardo. And Everardo, you may begin when you are ready.
Thanks, Deanne. And thank you very much for all of you attending this webinar. We hope you enjoy it. So the task today for myself and Mark is to argue that personalized cancer medicine needs randomized trials. I will first start by making a very brief introduction on personalized medicine. Then I will show you what appears to us to be a trend for perhaps increased use of non-randomized evidence, including for drug approval. I will talk a little bit about the debate between randomizing or not, but this will then be expanded on by Mark, who will tell us the statistical perils of non-randomized evidence, and he will argue that randomization is necessary or it may be used in phase 1, 2, and 3 trials. And then he will make some closing remarks with some tentative conclusions. So as we all know, oncology trials are changing, and the change appears to be more and more rapid. There is, of course, increasing knowledge on the biological features of cancer and an increased availability of many targeted therapies. There is also a change in regulatory environment, and perhaps these are the three key components of the changing face of oncology trials. There have been some recent approvals that were based not exclusively, but to a large extent on phase 1 data. And the examples that are often cited are crizotinib and ceritinib. The two drugs are tyrosine kinase inhibitors that may be used in non-small cell lung cancer and other malignancies. So crizotinib was approved based on a phase 1 study and also on a phase 2 study. So as you can see, the total number of patients that led to approval was not so large when compared with regular approvals. Now, of course, this was an accelerated approval. Then the full approval was granted 2 or 3 years later based on a randomized trial that had PFS as primary endpoint. But initially, the approval was based on non-randomized evidence and very, very early clinical experience with crizotinib. And the same was true for ceritinib. Now, this is not new if we look at the history of drug approvals. For example, the FDA publishes periodically an update of their experience with approvals. And in this paper, for example, between 2005 and 2007, of the 53 indications that are described, 37 approvals were based on randomized studies, which means that the other 16 approvals were based on single-arm studies, typically phase 2 trials. So the fact that drugs may be approved based on single-arm studies is not new per se. What's really changing is personalized oncology, which is, of course, an evolving paradigm according to which drugs are used in a very targeted way to in patients who have the correct features, for example, biomarkers. So this has been explained in different ways, and some people refer to personalized oncology as precision oncology. These two terms are equivalent. We just prefer to use personalized as a preferred name. So essentially, there are two ways to think about the use of targeted therapy and biomarkers. One is a traditional way in oncology, according to which we look at tumors depending on their histological origin, and then we look for some kind of marker or molecular abnormality and treat patients with some targeted therapy. And of course, in this example, the response rate is very low. It's not necessarily so in all situations, but in this example, a contrast is being made between the old approach and the new approach, according to which molecular alterations are more important, perhaps, than histology, and then you can treat patients with different targeted therapies regardless of the tumor type that they have, as long as they have the correct alteration. And hopefully we will get improved responses and more benefit from this kind of approach. Now, the typical example between of the difference between targeted therapy and chemotherapy, which is sort of the old paradigm in oncology, is in non-small cell lung cancer. So more than 10 years ago, people were struggling to find which chemotherapy doublet was better than the other, and many studies were conducted. This is only one example, and there were really very minor differences between combinations, not a difference in this particular study. So chemotherapy was being given to all comers, and the treatment effect was not really there when you compared different agents. Now, you fast forward to more recent times, and then when you're able to give a targeted agent to patients that have the correct molecular alteration, you see a large treatment effect, for example, when compared with chemotherapy. So this is already an old example that many people are familiar with. Now, there is a difference between targeted therapy alone and personalized therapy. When tyrosine kinase inhibitors against EGFR came about, they were being given to patients without the knowledge that there was a specific predictive biomarker. And then this is just one of 4 very large studies in non-small cell lung cancer, again, in which gefitinib, for example, provided no benefit when it was added to chemotherapy and compared with placebo in patients who were not selected for the correct molecular abnormality. And again, the same study that I have just shown, if you select the correct patients, and you compare now the agent alone with chemotherapy alone, you have a large treatment effect. So this is what we're talking about. We're talking about personalized therapy and not just targeted therapy in general. Of course, the key element in personalized medicine is the drug biomarker pair. So ideally, we should be able to know which patients are more likely to respond by looking at and validating predictive biomarkers. And there are many examples in oncology, as you know, for example, rituximab and CD20, trastuzumab, and other anti-HER2 agents, and then several agents against BCR-ABL changes, the EGFR inhibitors, and many others. But of course, we all know that there are drugs which have been approved and which are targeted agents but have no biomarker known so far. So in that regard, these agents may not be termed personalized in the sense that we still don't know if we're giving them to the right patients. Now, there is evidence of different types favoring a personalized approach. For example, in two recent studies, authors have looked at two different settings. For example, among published phase 2 trials, they have looked whether a personalized approach might provide improved results when compared with a non-personalized approach. And regardless of the methodology used, they found that both for response rate for PFS and also for OS, for overall survival, the personalized approach always provides better results, at least in published phase 2 trials. These authors have looked at FDA-approved agents and found more or less the same thing, at least for response rate and for PFS. They found that a personalized approach was associated with improved results when compared with a non-personalized approach, again, comparing FDA-approved agents. Now, this image comes from a paper that is written by FDA members, and it's a very interesting paper about expansion cohorts. And I like this figure because it really depicts what's happening in cancer clinical research in a very condensed way. So historically, we had a more clear demarcation between phases of development. Although they are not shown here, it's pretty clear that pharmacology refers to phase 1, therapeutic exploratory refers to phase 2, and therapeutic confirmatory to phase 3. Then safety was always a concern, and efficacy was something that started to be assessed from phase 2 onwards, and this eventually led to approval. Now, more recently, things are changing in some ways. One of them is the sort of loss of a clear demarcation between phases. Although we still refer to studies as phase 1 or phase 1-2 or phase 2, they're becoming more similar in the sense that phase 1 studies are growing, phase 2 studies are often randomized, so there is a less clear demarcation. But more importantly, perhaps, there is a growing trend to assess efficacy early on. So this is one of the major differences between before and now. And this is somewhat motivated by regulatory changes as well, with accelerated approvals. But of course, the chief driving force is personalized medicine again. This somewhat busy picture shows the schema for a study that not many people would think is a phase 1 trial if this slide was shown a few years ago. But now everybody knows that this is of course, the phase 1 trial for pembrolizumab, a very successful checkpoint inhibitor that was approved on the basis of, or at least received the breakthrough designation on the basis of the phase 1 trial that's shown here. So this study started out as a regular phase 1 study and had an initial cohort of only 30 patients. But then, based on the responses that were seen and on the need to assess several dose levels and different disease histologies, eventually the study reached an accrual that is, to my knowledge, the largest in phase 1 ever in history of 1,235 patients. So our question here is this a new model for phase 1 trials? And Mark will try to answer this question in his part of the talk. Now, the previous figure had many expansion cohorts, and we'll talk more about that. And here I just wanted to make the point that what have been called expansion cohorts, in other words, patients that are still in phase 1 but that are selected usually on the basis of histology are treated still within the phase 1 study to get more information about safety and efficacy and hopefully to get accelerated approval. And it's clear that the use of expansion cohorts has increased recently. There is a time trend for this, and also that this is clearly related to non-cytotoxic therapies, most of which are, of course, targeted and personalized therapies. Now, phase 2 designs are also evolving, and here I just show two prototypes, so to speak, of the novel phase 2 designs. There are others, but these two ones are the most talked about, at least. This is the so-called umbrella trial, according to which patients with a certain tumor type, for example, lung cancer, are profiled in terms of their genetic landscape of the tumor, and then they may be treated with different drugs according to the genetic profile. Now, there may be a role for randomization here, and I will show you an example in the next slide. Here, this example is with single drugs, but these studies may also be randomized. Whereas here, the so-called basket trials have a different approach. You can include patients in a so-called agnostic fashion with regard to their histology, and then what counts is the molecular analysis that will lead patients to be treated with different drugs. So the main difference here is that you have patients with the same tumor type, whereas here you have different tumor types, and what really drives the study is the molecular signature, so to speak. So if you look at these two types of studies as matrices, for example, here you have the tumor of origin, and then you can classify them according to several molecular subtypes, which is not new in oncology. This has been done since at least when microarrays came about 16 years ago. But whereas here you look at specific molecular abnormalities or pathways, and then you can sort of tell how many tumors have specific abnormalities. So these are really two different ways of looking at the same thing, taking into account biomarkers. And this is the battle trial, which is a prototypical, to my knowledge, the first umbrella trial in which the protocol specified that patients with lung cancer would undergo assessment of the molecular profile, and then they would be treated with different agents, not on the basis of the profile. They would be actually randomized to different agents, and then in retrospect, the results would be assessed according to which agents they received and whether they had or not certain molecular alterations that were sought in the tumor. And this has been reported for several different alterations. So this is one example of an umbrella trial. Whereas this is a prototypical basket trial. This was published recently, and the trial assessed vemurafenib for patients with various tumor types as long as they have the same BRAF V600 mutation. So here again you can analyze patients separately in cohorts. For example, this is the non-small cell lung cancer cohort, and the waterfall plot shows their response to vemurafenib. You can have a look at that side by side, for example, with colorectal cancer, which doesn't seem to be looking so good here. And then there is also other tumor types, because they allowed various histologies that were not in large enough numbers to make specific plots. So this is an example of a basket trial. And nowadays there are basket trials also in phase 3. This, of course, is phase 2. Now, personalized medicine is very compelling, and I say that as a medical oncologist, because when you look at a survival curve like this, you see that patients that had some kind of genetic driver identified and were treated according to the alteration fared much better than patients who had a certain molecular abnormality but got no targeted therapy, and so this fared as bad, so to speak, as those that had no driver abnormality identified. So this is very compelling, and we tend to conclude that targeted therapy, or in this case, really personalized therapy, is good. But of course, the problem here is that this is a non-randomized study. This is a cohort study. In a way, it's a retrospective analysis, and this begs the question of whether is rationale enough. And this is an old example, and it's not about targeted therapy the way we look at it today, but in those times there was very strong evidence that correcting anemia in head and neck cancer patients, and this was done with erythropoietin, would improve the outcomes of radiotherapy, and this was being done sort of based on rationale. And in fact, when a randomized trial was done, and this is survival, it's not indicated here, but this is overall survival, there was indeed a difference, a significant difference, but it favored placebo and not the agent that was thought to improve the outcomes. So we have to do studies in order to tell whether a certain agent differs from another one in the same patient population, and that means randomizing them between two different interventions. And now bringing the example to molecular targeted therapy, or in this case personalized therapy, we have the recently published Shiva trial, which provided results that were not so encouraging for the oncology community, because here patients with no standard therapy and who had received all the approved targeted therapies for their particular histologies were again profiled and then randomized to a targeted therapy that was based on the alterations that were found, or to treatment at physician's choice, which was essentially chemotherapy. And the primary endpoint of the study was progression-free survival, and there was no difference to speak of in terms of PFS. So this was sort of disappointing, and makes us question once again whether non-randomized evidence is reliable and whether historical comparisons are useful. Let's not forget toxicity, because as oncologists, we tend to think, and we're probably right in many cases, but we tend to think that targeted therapy is less toxic. It's probably true for some drugs, but not necessarily for all the drugs. So, for example, in the Shiva trial, at least nominally, the incidence of grade 3/4 toxicity was even larger with targeted therapy than with chemotherapy, and this is sort of also unexpected. But this is a randomized trial, although there have been criticism about this study. In the end, it's a randomized trial, and this kind of evidence, of course, sits at the top of the evidence-based medicine pyramid. Now, the problem, I guess one of the central problems and one of the reasons for thinking about this webinar and this whole story of randomization in personalized medicine has to do with ethical concerns that may be raised. And this was, for example, voiced in this editorial in the New England Journal of Medicine. So if you have a study, an early phase study that shows very promising results, how can you randomize patients, for example, in a phase 3 trial of that agent, if they know that the results have been so promising. If you have a highly targeted therapy that works very well in early phase studies, and you have patients that know that they have the same molecular alterations, they of course will want to receive the agent, and then conducting studies like large phase 1 studies may actually preclude running randomized trials down the road. And so this is where I will stop my part of the talk and hand it over to Deanne first for a poll, and then to Mark that will continue with the webinar.
Thank you very much, Everardo, for that portion of the presentation. We do indeed have a polling question for our audiences, and I'm going to launch the polling question on your screen right now. And for this polling question, you're able to vote more than one answer, and the poll is: randomization is not required to test agents for outstanding efficacy, a nice-have rather than a must-have, only useful in phase 3 trials, and the last option is always useful in trials of all phases. And again, for this polling question, you can vote more than one answer. And if you have any other questions or comments you may have for our first speaker, you could just do so in the questions chat box, and we'll try to attend to your questions during the Q&A session. And it looks like some of you have voted. I'm just going to keep the polling running for a bit more so that most of you can vote. And again, the polling question is: randomization is, and all the 4 answers that you can vote more than once. Okay, looks like most of you have voted. I'm just going to close the polling question right now, and I'm going to share the answers with everyone. And looks like we have a 92% of you says always useful in trials of all phases, followed by a 24% of a nice-have rather than a must-have, followed by 18% that said not required to test agents of outstanding efficacy, and a small 2% saying only useful in phase 3 trials. Great, and now without further ado, I would like to hand over the mic to our second speaker, Mark. And Mark, you may begin when you are ready.
Thank you, Diane. This poll was kind of an interim analysis in this webinar, really, and I think Everardo and I sort of decided that if the answer would be 100% of the participants chose that, randomization was useful in all phases, we might as well stop the webinar now, because that is exactly what we try to show. And so 92% of the participants voted for that option. So our job is almost done, not quite, but almost done, because we want to convince everybody that randomization needs to be at least considered in planning clinical trials in oncology right from phase 1. And that is what I'm going to try to demonstrate in the remainder of this webinar. So of course the debate about randomization is not new. There's been a sort of historical debate about randomization from the very time it was proposed in the 50s. These are interesting papers that going back to the 70s. The first one was by Tom Chalmers, who was an MD, not a statistician. And surprisingly enough, that paper is now 45 years old, and it argued that when you try a new drug, the only way to do it ethically is to randomize the first patient, because Tom Chalmers and his colleagues argued that you may not ethically deprive a patient from the potential benefit of a treatment that is already known and expose this patient to a new drug that may come with toxicities and no efficacy. So this is kind of an interesting historical perspective, because what we see today is exactly the same as what Chalmers and colleagues described 45 years ago. We still have this problem of exposing patients to a drug that's not known at all. Maybe the preclinical evidence is compelling, maybe the rationale is compelling, but you still don't know when you expose the first patient how the patient is going to respond to this drug. And particularly when you have targeted agents or now immunotherapies, which really fool around with the immune system, I think there's a lot of unknowns there. And the Chalmers argument that you may not ethically deprive the patients from the benefit of a known treatment, I think remains as valid today as it was 45 years ago, regardless of personalized medicine or precision oncology. Of course, another paper appeared soon after in 1974 by Guillen and Freireich, a statistician and a clinician, and they argued that you can probably use non-randomized controls in cancer clinical trials by trying to match historical data from patients who had been treated before with new patients that come in the study today. And I think by and large this paper has been proven to not apply because and I will come back to this in a moment, the dangers of trying to match patients far outweigh the benefit of doing so. And then, the clinical trials, of course, continued to be randomized, including in situations where it was very, very difficult to randomize. And one example that comes to mind, it's a celebrated example, is the first trial of AZT in the treatment of patients with AIDS and AIDS-related complex back in the 80s, where, as you recall, AIDS was a lethal disease, and AZT was really the only drug that was around. There was absolutely nothing else to give to these patients. They would die of AIDS pretty soon. And so this was a dramatic situation where a randomized placebo-controlled trial was carried out. And actually, in retrospect, this may have been the best strategy because AZT was very soon seen to be extremely efficacious. At one point, the interim analysis was shown to an IDMC with 11 deaths in the placebo group and 1 death only in the AZT group, and that was sufficient for the IDMC to recommend stopping the trial. And here you see the final outcome of this trial published a few years later, where it was very clear that AZT as an antiretroviral drug was very efficacious. And that led, of course, to the developments we've seen since, where patients today have a very good prognosis, even though they would die in those days of their disease. So again, a celebrated example, but this issue about randomizing versus not randomizing comes back on a regular basis. And there are papers sort of arguing that you should randomize, but more importantly, there have been recent papers where these authors in fact argued that you need more rather than less randomized trials if you now talk about personalized medicine. So in a sense, what they argue is the exact opposite to the trend that Everardo mentioned before, which is that with personalized medicine, people are very tempted to forget about randomization. And these authors, I think, successfully argue that it's, if anything, is the exact opposite. And again, even today, for very dramatic diseases like Ebola, the Ebola epidemic, of course, is a very serious condition, perhaps similar to AIDS 20 years ago. And yet, the need for randomized trials in Ebola has been argued for. And so why do people argue so strongly that you need randomized evidence? Well, non-randomized evidence is very dangerous to base any decision on because of these factors here. Selection bias. Patients who enter clinical trials are not a random selection of all potential patients. They are very carefully selected. That's the role of the oncologist, is to decide what patients can go on a phase 1, a phase 2, or a phase 3 trial in cancer. And so selection bias plays a major role here because there are so many unknown confounders. There are so many factors that we know very little about that drive the outcome of a patient in addition to the treatment they receive. Obviously, there is stage migration, which makes historical comparisons very hard. We all know this, that patients tend to do better today than they were doing 20 years ago. And of course, treatment plays a major role here, but there is also better detection and earlier detection of cancer. So there is stage migration to earlier stages. And there is trends in supportive care. Again, the patients are better treated today, even in terms of supportive care than they were 20 years ago. And this is, of course, very good, but it makes historical comparisons very unreliable. And finally, the populations of interest are, of course, patients with early disease, and most typically the sort of early trials like phase 1, phase 2 will, in fact, be trials where patients have advanced disease. And so for all this reason, it's very hard to use non-randomized evidence from advanced disease to infer anything about the treatment effect in earlier disease. And in contrast, the benefits of randomization, they are all well known. I won't belabor these points, but it's just useful to remind ourselves of the benefits. The control of selection bias is, of course, the big reason we want to randomize. We have a control, an internal control for all efficacy and safety outcomes, including the benefits on sort of important clinical endpoints like overall survival, for which you can't expect very, very large benefits. You can expect large benefits of targeted treatments on endpoints such as responses or even perhaps progression-free survival. But for overall survival, the benefits are bound to be much smaller. And of course, to make sure that these benefits are real, we do need relatively large-scale randomized trials. And then something that I will get back to as well is we often want to validate predictive biomarkers or even prognostic biomarkers. We don't need randomized trials to validate prognostic markers, but we do need randomized trials to validate predictive biomarkers. And these are more important with personalized medicine than they ever were before. Hence the reason for randomizing patients, even for targeted agents and for personalized oncology. And finally, if we want to validate surrogate biomarkers, here too, we do need randomization. So let me come to some important issues to remember when we discuss the need for randomization. First off is even for agents that have been very successful in terms of inducing responses or improving progression-free survival, like crizotinib here versus chemotherapy. In fact, the benefit of these agents on overall survival, which is after all the endpoint that matters most, may be much less convincing. And again, had we not run this randomized trial here comparing crizotinib with chemotherapy, we actually wouldn't know what the real benefit of crizotinib is on survival. Now, obviously, the lack of a survival benefit may be due to a variety of reasons. We won't discuss these today, but there are very good reasons why survival may not differ. But it's very important to quantify this, the lack of benefit or the benefit of a new drug on survival. And so we do need to do randomized trials to know this. The other important consideration is what do we want to randomize? And of course, what we have in mind is the sort of obvious randomization of new drug experimental versus standard of care. This is the most obvious way that we could randomize patients between some standard of care, if one exists, versus the new drug. But there are many other ways we can randomize patients. We could do, as in the Shiva trial, experimental versus physician choice. Regardless of what physicians prefer, we will randomize patients between that choice, whatever it may be, and a new agent. We could also use the add-on designs. In other words, all patients receive the standard of care, which is appropriate for their condition, plus or minus the experimental agent. Or we could even do physician's choice, plus or minus the experimental agent. Now, all these cases would really randomize the new drug versus some kind of control. But if that's difficult to do for any reason, then we can also think of randomizing patients between the single new agent versus a combination of that new agent plus something else that might be appropriate. We could even think of randomizing doses, perhaps including a very low dose, and this is something the FDA typically likes, is that you randomize patients between several doses, one of which is so low that it is probably inactive, not surely inactive, but probably inactive. So this almost serves like a control group. You can randomize between several schedules. Very often there are questions as to how many times the drug needs to be given, or at what dose and how frequently. You can also randomize the duration of the drug. In other words, this would be a randomized discontinuation design. And we can finally randomize a drug to be given immediately versus to be given later. This is often called a window of opportunity trial, where a patient is not in such bad condition that they can actually be left untreated for a little while, not too long obviously, a little while, and then be treated with a new agent. And then of course randomizing immediate versus delayed provides a lot of data that can be quite useful to assess the true efficacy of the new drug. Evrada already showed this slide before. This is the phase 1 trial of pembrolizumab, and you didn't see it perhaps because this is a heavy slide, but I've highlighted in pink here the fact that some patients were indeed randomized in this trial, but they were randomized without a control group. They were randomized between different doses of this new agent. So this is, in a sense, the lowest version of randomization, where you randomize the patient to different doses of the same agent. And obviously, that is not the best way to learn about the efficacy of the drug. a better way to learn about the efficacy of the drug would have to be with a standard of care control. But what is the appropriate control? Sometimes best supportive care, which means essentially nothing, is the appropriate control, but this is less and less the case, and fortunately so. So we have less and less situations where patients really have no treatment option left, except, of course, if you think about last line treatment, but with more and more drugs being available, these situations become less and less frequent, that there is absolutely nothing that can be given to the patient. Another appropriate control is any treatment other than the experimental treatment that the patient would receive if they were not entered in the trial. So in fact, we have a very wide range oftentimes of treatment options that could be considered. And the Shiva trial provides a nice example of this, where the randomization was between anything that the physician wanted to give, and that might depend on a number of different factors, including molecular alterations, versus a targeted treatment. So in a sense, the Shiva trial, again, was a very flexible design where the patients could receive something that was according to the physician's choice. And this included all treatments that have been reported to improve survival of quality of life in randomized trials. Another important consideration to remember when we plan a randomized trial in early disease, in early phase trials, phase 1 or phase 2, is how can we randomize patients in such a manner as to learn as much as we can about the new agent. And very often a randomization ratio or an allocation ratio of 2:1 or sometimes even 3:1 is favored. And that means that the experimental treatment has a higher likelihood of being given than the control group. Now, I think it's very important that this cannot ever be justified on the basis of ethics. It's a common misconception that it's ethically better to submit more patients to the new agent than to the control group because the new agent is very promising. If you are truly in equipoise, there is no reason to submit more patients to the new treatment. But the reason to do so is that it provides more information on the treatment that's not known and less information on the treatment that is already well known from past experience. So in a sense, this design is justified just based on information contents rather than on ethics. A point that I wanted to make in passing, but it's an important one, is that in our opinion, allocation should not be adaptive. And what we mean by adaptive is that there are some trials in which the treatment was given to patients according to the results of previous patients. And the allocation favored the better performing treatments. So you would look at how many patients were already in the trial and how many had responded to the new treatment versus the control. And you would actually favor either the new treatment or the control, whichever one did better. This actually is not desirable for several reasons, but the two main reasons is that it's very hard to interpret these trials statistically. It's a mess, but it also may lead to more patients receiving the worst treatment, paradoxically. So these trials, even though they may appear to be preferable, may actually turn out to be worse than just a fixed allocation randomization. The battle trial that Everardo already mentioned actually used this trick. They randomized patients between 4 treatment arms, depending on their biomarker profile here. And in fact, they used an adaptive allocation such that either one of these drugs was favored depending on the results of the patients already entered. Again, something that may seem to be smart and interesting, but in fact is not. This is a good reference if you're interested in this particular aspect of randomized trials, the adaptive randomization. This is a paper by Korn and Friedlin in which they recommend using a fixed randomization, 1:1, 2:1, 3:1, doesn't matter, but not an adaptive randomization. And finally, the ethical aspects of adaptive randomization trials have been discussed in great detail in this issue of the journal Clinical Trials, for those of you who are interested. What about stratification? More and more we now have information about the patients that may be key in interpreting the results of a trial, particularly molecular alterations that may make the patient susceptible or not susceptible to some new agents. And of course, it's important then that the treatment allocation be stratified for known or potential prognostic and predictive factors. Then one technique to do this, which works really well, if you have several factors that you want to adjust in the randomization schedule, including the center, is a technique that's called minimization. And contrary to what many people believe, you can actually factor in a lot of different factors at baseline if you use minimization, even in relatively small trials. And of course, the benefit of balancing the randomization is particularly acute in small trials where you don't have the luxury of the large numbers, which will tend to make everything balanced anyway. So for small trials, phase 1, phase 2 randomized trials, it is of interest to look at minimization as a proper way to allocate patients to treatment. And finally, let me turn to the role of biomarkers. As we know, there are two types of biomarkers, which are often called prognostic and predictive. Prognostic biomarkers are simply biomarkers that have an impact on the prognosis of the patient. So, for example, here, progression-free survival is depicted for patients with EGFR wild type and for EGFR mutant. And we see that EGFR mutant patients do better than patients with wild type tumors. But that is, of course, of limited interest because what is really important is predictive biomarkers, biomarkers that predict the benefit of some drugs. And here, you remember this slide, again, Rado showed it, is gefitinib versus standard chemotherapy for patients with non-small cell lung cancer. And it's obvious that patients who received gefitinib did better if and only if they had a mutation of EGFR. So in a sense, this trial alone, this randomized trial alone in patients with a mutation wouldn't be sufficient in order to show that this drug is indeed better, gefitinib is better than chemotherapy in EGFR mutated patients. You would also need to have the wild type patients, and this was done in a trial which is called the IPASS trial, done in Asia, where it was obvious in the same randomized trials that patients who had the mutation benefited from gefitinib, and patients who had the wild-type tumor did not. Actually, these patients were deprived from the benefit of standard chemotherapy, and therefore they did actually significantly worse than patients with standard chemo. And so this is the perfect example of a predictive biomarker where a mutation of this gene would actually predict the benefit of a targeted agent. And so this is important because it was only in this randomized trial context that it was completely clear that EGFR mutation was indeed a predictive factor for the benefit of gefitinib. Now, the problem is that, as I said, we look at interaction between the treatment effect and the presence of this biomarker. Let's call it biomarker plus or minus. The problem with this is that statistically we know that interaction tests have very low power. And so, in a sense, oftentimes we can't reach significance for an interaction, even if it's very strong, because we lack sensitivity or we lack power to detect these interactions. And the hypothesis of an interaction may not even be relevant. And this leads into the clinical relevance of interaction. I'm going to show with an example that the situation is not as simple as we would like. Assume that we have a biomarker and we call the two levels of the marker BM plus and BM minus, so patients can have the biomarker positive or biomarker negative. And let's assume that we have a treatment that has an effect of a hazard ratio HR of 0.4 in one of the subsets and 0.7 in the other subset. So there is a significant interaction. The hazard ratios are not the same in the two subsets. So based on the example of the IPASS trial, should we treat one subset and not the other? Well, it depends. Suppose that the biomarker is also prognostic. It's not only predictive, it's also prognostic, such that biomarker positive patients do far worse than biomarker negative patients. Suppose that the hazard ratio of 0.7 is seen in the biomarker positive patients, and the hazard ratio of 0.4 is seen in the biomarker negative patients. Now, I think any oncologist would say this is a huge and very worthwhile effect for these patients. You reduce by more than half their risk of an event. But this is a benefit, even though this is a hazard ratio of 0.7, which may look impressive in relative terms. In absolute terms, these patients do badly. Even if you treat them with this agent, maybe the agent is not worthwhile for BM plus patients. Whereas, in contrast, if the hazard ratio of 0.4 applied to the biomarker positive patients, well, then that would be a worthwhile difference for sure. And in the biomarker negative, of course, the effect now is much less because the hazard ratio is 0.7 in relative terms. But in absolute terms, these are patients of really good prognosis, and you still can improve their prognosis by something that may not be insignificant, clinically speaking. So here the treatment might be recommended for all patients. And so clinically, the interaction that we test statistically may not be the prime consideration for treating or not treating patients. And so it's very important, again, to have randomized evidence on these interactions. And this is only possible with randomized trials. So if we want to validate biomarker trials, if we want to validate a predictive biomarker, for example, a gene mutation, well, randomized trials are needed, whether you like it or not. You do need randomized evidence. Worse than that, you need randomized evidence on the biomarker plus and the biomarker negative patients, which is even worse because in one case, biomarker positive patients might benefit and biomarker negative patients might not, but you need randomized evidence on both subsets. And large-scale randomized trials are needed because interaction tests lack power. So this is a sequence of bad news, really. We do need randomized trials, and not only that, we need large-scale randomized trials in order to fully validate our biomarkers. And if we're interested in surrogate biomarkers, it's even worse because then we need randomized trials of large scale, but we need several randomized trials in order to fully validate a surrogate biomarker-based endpoint such as circulating tumor DNA to confirm that the treatment effect on the surrogate, let's say the circulating tumor DNA, is predictive of the treatment effect on the true endpoint, perhaps survival. And so we do need randomized trials and large randomized trials, and many of them, in order to fully validate the biomarkers that we may be interested in. So let me conclude. Oncology trials, as Rado showed, undergo profound changes and chiefly driven by personalized medicine. Improved outcomes in a single-arm trial may be due to selection bias or prognostic impact of these molecular alterations that are now better determined. The predictive impact of these alterations cannot be validated without randomization, and therefore randomization is essential at all stages of development. And I'm so pleased that many of you already believe this to be the case before the webinar, or at least at the time of the polling question, and hopefully that percentage of patients hasn't gone down by now, but has gone up. We need randomization in phase 1 as soon as the expansion cohorts kick off. I don't think it's reasonable to do a 1,200 patients trial in phase 1 without randomization, or at least without randomization to a proper control. The proper control is often the standard of care. If standard of care is not available, then you can look at different ways of randomizing patients. And of course, we must always have early stopping rules in our trials, regardless of the phase. In phase 1, we could stop for outstanding activity based on biomarkers. Phase 2, again, we want to select regimens more likely to succeed, and we need a randomized trial to do this. Whenever indicated, we can include assessment of biomarkers, and again, we have stopping rules for outstanding efficacy such that the trial doesn't become unethical if indeed the new drug has outstanding efficacy. We can use these intermediate endpoints for these interim analyses, such as response rate. And finally, we need, obviously, randomization in phase 3. For those trials, we need a true control, which is the best treatment patients would receive outside of the trial, possibly nothing, actually, but we need a true control. And again, we need to have early stopping for outstanding efficacy. And I think we need to have more aggressive interim analyses in our clinical trials to continue to have ethical trials, in spite of the fact that we randomize patients. Thank you very much for your attention, and now on to you if you have any questions that you would like to raise and to discuss with us.
Thank you, Mark, very much for your portion of the presentation. We have come to the Q&A portion of the webinar, and we do have one question, or a couple of questions coming from the audience, and we have one right here for both our speakers. And one coming in from the audience is, the FDA has approved many drugs with non-randomized studies. Can you give us a summary under which situations randomization is not required?
Boy, that's a tough one. That's a tough one. Well, I think the problem is that the cases we know of where drugs were approved based on non-randomized evidence, let's say pembrolizumab, that was really a stroke of luck. I mean, that drug had outstanding efficacy, and in retrospect, that was an approvable drug because there were so many responses never seen before in this patient population that the FDA was really compelled to approve it based on that particular trial. But the thing is, if you now plan a new trial for a new drug, is it reasonable to assume that that new drug will have outstanding efficacy? I don't think that's a reasonable assumption to make in general. And quite frankly, if the pembrolizumab trial had been randomized, I think it would have made the life of FDA and even of the company far easier, because they would have had compelling evidence right from phase 1 that this drug had activity, and they could have continued the randomization in some patient populations to prove that the drug not only had outstanding efficacy on tumor response, but that it also improved survival of these patients. So I think if you can randomize earlier, coming back to Chalmers' ID, randomize from the first patient, you can set yourself up to actually have a much faster full approval of the drug rather than just conditional approval. Because when you get conditional approval for a drug of outstanding efficacy, you still are left with the question that the FDA and the EMA will answer, which is, prove us that this drug works in most patients, not just in the highly selected patient population you had in phase 1. Prove us that the drug does something truly beneficial to these patients and all of them, not just a very highly selected group. That would be my reply, but Everardo, you may have a better oncological reply to offer.
I wish I had. That's a question we struggle with. Are there situations where we can really forego randomization? Do you have a situation where you have such compelling evidence in a very selected population, and then it's acceptable not to randomize? And it's very hard to come by with an answer that is always valid. I think nobody knows yet. But if I may add something, it's not really a direct question. There has been a recent sort of an editorial in the New England Journal signed by FDA officials, and they describe the experience with pembrolizumab, and they talk about the seamless development in oncology. And my understanding of the paper, which is quite short, it's a short piece, it's that they're not really encouraging people to repeat the pembrolizumab experience or development schema without very close interaction with the FDA, and preferably, and I think that's the take-home message of that article, is that preferably the drug should receive breakthrough designation before it can be considered for a phase 1 study like that one. So this may be of interest to some people in the audience to read that paper, because it shows the thinking of the FDA about this.
So what you're saying, Everardo, if I understand, is that you do a phase 1 trial if you have outstanding proof of efficacy, or proof of outstanding efficacy, rather. You go to the FDA, discuss breakthrough designation, and discuss with the agency what trials designs would be appropriate for the remainder of the development.
Yeah, I guess more explicitly, they don't want to encourage people to try and get approvals based on phase 1 studies like, it happened for other drugs. One of them is pembrolizumab, is the example that they use. I think they still want people to be more, in a way, more conservative, and despite having a seamless transition, be more careful and not consider that every drug with promising activity will be approved on the basis of a large phase 1 study, unless it's really a breakthrough designation. That's my understanding.
Well, thank you very much for those answers. We have reached the end of the question and answer portion of this webinar. If you have further questions, please direct them to the email address showing on your screen, and that's dominique.grissard@iddi.com. Now, please join us in thanking our speakers, Dr. Everardo Saad and Mark Boyce. Thank you both.
Thank you.
Thank you. Thank you, everyone, for participating in today's conference. You will be receiving a follow-up email with access to the recorded archive of this event, and a survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our future webinars. We hope you found this conference informative. Have a great day, everyone.